Good day, everyone. How are you? Today, we will be discussing our Session 3 genetics class. The Mendelian inheritance in humans. Although many human traits clearly run in families, most do not show a simple Mendelian pattern of inheritance. Suppose, for example, that you have brown eyes, but both your parents' eyes appear to be blue. Because blue is normally considered recessive to brown, does this mean that you are adopted or that your father isn't really your father? Not necessarily, because eye color is influenced by more than one gene. In contrast, single gene traits in people usually involve an abnormality that is disabling or life-threatening. Examples are the progressive mental retardation and other neurological damage of Huntington disease, and the clogged lungs, and potential respiratory failure of cystic fibrosis. A defective allele of a single gene gives rise to Huntington disease. Defective alleles of a different gene are responsible for cystic fibrosis. There were roughly 4,300 such single gene traits known in humans in 2009, and the number continues to grow as new studies confirm the genetic basis of more traits. Now, let's take a look at how these traits are handed down from one generation to next. Pedigrees aid the study of hereditary traits in human families. What do we mean by pedigree? It is an ordered leaf diagram of a family's relevant genetic features, extending back to at least both sets of grandparents and preferably through as many more generations as possible. Determining a genetic defect's pattern of transmission is not always an easy task because people make slippery genetic subjects. Their generation time is long, and the families they produce are relatively small, which makes statistical analysis difficult. They do not base their choice of mates on purely genetic considerations. There are thus no pure breeding lines and controlled matings, and there is rarely a true filial second generation like the one in which Mendel observed the 3:1 ratios from which he derived his rules, because brothers and sisters almost never mate. Figure 2 shows how to interpret a family pedigree diagram. The square represents male, circles are females, diamonds indicate that sex is unspecified. Family members affected by the trait in question are indicated by a field symbol. A single horizontal line connecting a male and female represents mating. A double connecting line designates consanguineous mating, which means a mating between relatives, and a horizontal line above a series of symbols indicate children of the same parent, or sibship, which is arranged and numbered from left to right in order of their birth. Roman numerals to the left or right indicates the generations. A vertical pattern of inheritance indicates a rare dominant trait. Before we will discuss the figure shown to you, let us take a note on these following terms. Heterozygote. It refers to an individual having two different alleles of a particular gene. This pair of alleles for Huntington disease is present in a heterozygote individual. Homozygote. This refers to an individual having the same allele of a particular gene. This is an example of an allele in a homozygote individual. All individuals represented by filled-in symbols are heterozygotes, except for this one who could have been homozygous for the dominant Huntington disease allele. All individuals represented by open symbols like this one are homozygotes for the recessive normal allele. Among the 14 children of the consanguineous mating, DNA testing shows that some are homozygous dominant, some are heterozygote, and some are homozygous recessive. The diamond designation masks personal details to protect confidentiality. Now, let's take a look on horizontal pattern of inheritance, which indicates a rare recessive trait. Unlike Huntington disease, most confirmed single-gene traits in humans are recessive. This is because, with the exception of late-onset traits, deleterious dominant traits are unlikely to be transmitted to the next generation. For example, if people affected with Huntington disease died by the age of 10, the trait would disappear from the population. In contrast, individuals can carry one allele for a recessive trait without ever being affected by any symptoms. Figure 2 shows 3 pedigrees for cystic fibrosis, the most commonly inherited recessive disease among Caucasian children in the United States. A double dose of the recessive allele causes a fatal disorder in which the lungs, pancreas, and other organs become clogged with a thick viscous mucus that can interfere with breathing and digestion. 1 in every 2,000 white Americans is born with cystic fibrosis, and only 10% of them survive into their 30s. Mendel answered the 3 basic questions about heredity as follows. What is inherited? He replied, alleles of genes. How is it inherited? According to the principles of segregation and independent assortment. What is the role of chance in heredity? For each individual, inheritance is determined by chance, but within a population, this chance operates in a context of strictly defined probabilities. Within a decade of the 1900 rediscovery of Mendel's work, numerous breeding studies had shown that Mendel's laws hold true not only for 7 pairs of antagonistic characteristics in peas, but for an enormous diversity of traits in a wide variety of sexually reproducing plant and animal species, including 4 o'clock flowers, beans, corn, wheat, fruit flies, chickens, mice, horses, and humans. Some of these same breeding studies, however, raised a challenge to the new genetics. For certain traits in certain species, the studies uncovered unanticipated phenotypic ratios, or the results included first filial and second filial progeny with novel phenotypes that resembled those of neither pure breeding parent. This phenomena could not be explained by Mendel's hypothesis that for each gene, 2 alternative alleles, one completely dominant, the other recessive, determine a single trait. We now know that most common traits, including skin color, eye color, and height in humans, are determined by interacting between 2 or more genes. We also know that within a given population, more than 2 alleles may be present for some of those genes. Now, this shows how the genetic analysis of such complex traits, that is, traits produced by complex interaction between genes and between genes and the environment, extended rather than contradicted Mendel's laws of inheritance. How do we recognize dominant and recessive traits in pedigrees? So this too represents the dominant traits and the recessive traits. The extensions to Mendel's laws. Unlike the P traits that Mendel examined, most human characteristics do not fall neatly into just two opposing phenotypic categories. These complex traits, such as skin and hair color, height, athletic ability, and many others, seem to defy Mendelian analysis. The same can be said of traits expressed by many of the world's food crops. Their size, shape, succulence, and nutrient vary over a wide range of values. Lentils, or lens culinaris, provide a graphic illustration of this variation. Lentils, a type of legume, are grown in many parts of the world as a rich source of both protein and carbohydrate. The nature plants set fruit in the form of diminutive pods that contain two small seeds. These seeds can be ground into meal or used in soups, salads, and stews. Lentils come in an intriguing array of colors and patterns, as shown in the picture, and commercial growers always seek to produce combinations to suit the cuisines of different cultures. But crosses between pure breeding lines of lentils result in some startling surprises. A cross between pure breeding tan and pure breeding gray parents, for example, yields an all-brown first filial generation. When these hybrids self-pollinate, the second filial plants produce not only tan, gray, and brown lentils, but also green. In this array of green, brown, and red lentils, some of the seeds have speckled patterns, while others are clear. Mendel's laws, then, they were assumed to be determined by a single gene with alternative dominant and recessive alleles. Many traits, however, did not behave in this way. For some, no definitive dominance and recessiveness could be observed, or more than two alleles could be found in a particular class. Other traits turned out to be multifactorial, that is, determined by two or more genes, or by the interaction of genes with the environment. The seed coat color of lentils is a multifactorial. Dominance is not always complete. A consistent working definition of dominance and recessiveness depends on the first filial hybrids that arise from a mating between two pure breeding lines. If a hybrid is identical to one parent for the trait under consideration, the allele carried by that parent is deemed dominant to the allele carried by the parent whose trait is not expressed in the hybrid. If, for example, a mating between a pure breeding white line and a pure breeding blue line produces first filial hybrids that are white, the white allele of the gene for color is dominant to the blue one. If the first filial hybrids are blue, the blue allele is dominant to the white one. As the figure shows, crosses between two breeding strains can produce hybrids with phenotypes that differ from both parents. We now explain how these phenotypes arise. That is all for this session. See you in the next one. Stay safe and God bless everyone. Bye!